morning, everyone. First, I'm going to introduce myself. I'm Sofiane Fraine from the Master of Biomedical Engineering, and I'm here today to talk about the characterization of femtosecond laser and crystalline lens interaction by biological techniques. First of all, I'm going to talk about the biolaboratory. It was founded by the Professor Philippe Guin and Professor Gilles Churet. The lab focused its research on the corneal graft engineering in association with Manutech, and also on the treatment of presbyopia in the collaboration with the startup Kejaco and Manutech. And the common point between BioMedical and Kejaco is to work on the femtosecond laser. Now I'm going to give you the background and objective of our study. So we work on presbyopia, though the question is what is presbyopia? Is a physiological process related to aging. And during this process, we have a loss of the elasticity for accommodation, as we can see in the picture. So in the clinical point of view, it results in close blurred vision. And now, in the biological view, our hypothesis is that the crystalline lens loss its elasticity in the nucleus that allows a rigidification of the lens. So the question is, how can presbyopia be treated? So we think in the lab that thanks to the femtosecond laser, we can restore the lens elasticity to restore the clear near vision. Recently, the lab have made some experiment on the interaction on the crystalline lens and the femtosecond laser, and we observe here the first result. We observe with different energy some bubble in the picture, but we have to develop some biological staining to observe the real effect of the laser treatment on the crystalline lens. And this is the bio-objective, to develop tools for the characterization of the laser treatment on the lens. This study is based on another study by Lim and al. Who work on a young human lens and not in the pig. There is no study on the lens fiber for pig and presbyopic human crystalline lens. And the objective of my internship was to observe lens fiber on the pig by biological staining from the epithelium to the center of the cut. To do that, we have two techniques, the chemical dye with the phalloidine and also the immunofluorescence. First, I'm going to talk about the phalloidine. Its benefit is quick observation and allows to observe lens fiber. As we can see here, we have the first result. We can see lens fiber from the epithelium to the center of the cut. And we can also observe the particular hexagonal shape of the lens fiber. So the phalloidine, it's a quick technique to observe lens fiber from the epithelium to the center of the cut. Now we are going to talk about the immunofluorescence. So its benefits allow to observe lens protein and fibers from the epithelium to the center, but its disadvantage is its long observation. First, we have tested a protein called aquaporin 0. Aquaporin 0 allows water to cross membrane, and we observe on the picture that aquaporin 0 have a stain from the epithelium to the center of the cryocut. So aquaporin 0 with phalloidine can be a great marker of the lens fiber from the epithelium to the center of the cut to observe the crystalline lens. The second one is the encaderine. Encaderine is a protein that allows cell adhesion. We can see that it is very located in the epithelium. This is a very innovative point. We have never seen this before. And we have also the observation that the encaderine is located in the cortex of the cut. But we can see in the picture 6 that we lost the stain. We think in the lab that the encaderine could be a great marker to discover the border between the nucleus of the lens and the cortex of the lens. So to conclude, presbyopia allows of the crystalline lens a loss of elasticity in the nucleus. So to characterize that, treatment laser, we have developed some biological staining with the coloration of the phalloidine and immunofluorescence techniques. And now we are going to characterize the real biological effect of the femtosecond laser treatment on the crystalline lens. And I want to say thank you for your time and your attention.